So the topic I'm going to cover is some of the causes of congenital heart disease, thinking more specifically about the genetic causes, and also to briefly introduce some of the emerging research which may be relevant, not right now, but into the future. We all know the important questions that are asked around the time of delivery, particularly if the diagnosis has not been known before that time. And probably one of the biggest questions there is, why did this happen? And we know what the things are behind that question. Yes, there's a need for information, but there's also a whole lot of other things which I will let Nadine Kasparian and Laura address this afternoon in understanding how we think and how we contextualize things. What we do know, though, particularly from the work from my PhD student Gillian Blue, who published the paper on the bottom right there, is that having an understanding of where this congenital heart disease comes from is actually very helpful in determining how you think about it and how you go forward in life as an individual and a family. And there's other reasons to focus on the genetics of congenital heart disease, simply because understanding why it occurs might also give us insights into how we can treat it more effectively using methods which are beyond the simple cut and sew that we have available to us at the moment. My short story is you should download this manuscript, also by Gillian. It's a few years old now, but it's still pretty relevant. This is available free from the Medical Journal of Australia website, accessible to everybody. And it goes through the causes of congenital heart disease, and it introduces both the genetic causes as well as the environmental causes. If you were to take a very quick snapshot, you'd say of 100% of congenital heart disease, we know the cause in about 20% of cases. And mostly this is due to individual gene problems with chromosomes, or interactions between those things and other problems in cardiac development. There are also some things that are labelled there as teratogens, which are really things that have been ingested or that you've been exposed to that might cause a problem. So in this group includes all the Down syndrome, the velocardiofacial syndrome, the Turner syndrome. But for 80% of other people with congenital heart disease, it comes apparently out of the blue. It's what we call sporadic heart disease. And through complex genetic analysis, we know that many people do have a strong genetic component to that, but that it's not easy to define what the genes are or what those genes are doing, or add a predictive risk having identified what those genes are. So it's a big blue box. Of the 20% of patients we do understand what the cause is, we look back to this what we call a transcription factor pathway. These are the genes that work in concert to turn a little tube of muscle into a fully functional heart in the first few weeks of the fetus's life. And whilst we can be very sophisticated in our analysis of mouse heart development, there's only so far you can translate that information to the human condition. Nonetheless, most of what we see still comes back to this transcription factor pathway, which is relevant later in the talk. When I mention some environmental risk factors, the most common environmental, I'm talking about environment for the fetus, is when mum has gestational diabetes, or even a greater risk is diabetes long-standing requiring insulin. That's probably the most common reason in our society that there's an environmental risk factor that may contribute to congenital heart disease development. Other things are rare: rubella, phenylketonuria, various inflammatory states, and medications are listed there. There are some things that people are concerned about, such as antidepressant medication, but the level of evidence for these at this stage is quite low. One of the biggest questions that we get is about recurrence risk. And we know that, I'm going to go through this as though it means something very special, but it really doesn't, because it's all generated from data from the 60s and 70s in population studies, populations that may be quite different to our population. Nonetheless, up until recently, this is all we've had to deal with. If we say the background risk is 6 per 1,000, just under 1%, the chances of another sibling being affected in the family are double that. It's still a very low risk. And you can, in some sorts of congenital heart disease, such as left heart obstruction, including mitral stenosis, aortic stenosis, coarctation, hypoplastic left heart syndrome, then the risk of the recurrence risk is higher for left heart conditions. It's also a little bit higher if a pregnancy is in a mum who has congenital heart disease herself. But when I say it doesn't really mean anything, none of this is personalised information. And really what we've tried to do is increase the amount of available personalised information. That's where research is going in this domain. So if we take out the known syndromes, that 20% of patients, and look at the patients that have congenital heart disease out of the blue, there's also this other group where there's quite a lot of congenital heart disease, but it isn't being passed down in a way that we can say that gene has caused that and it will cause that in their offspring. There's actually quite a lot of overlap between those two groups. And we used the DNA bank, which I know many of you who live in Sydney and have been treated at this hospital have contributed to work up an understanding of what the genes are that are relevant and to give us a direction towards finding personalised information, which I think is what you want. I put this slide up to illustrate the complexities, even when you know a gene and understand what it does. This is a very well characterised gene. It's called TBX20. It's in that pathway that I showed before. But even the same mutation in that gene can cause different patterns of disease in different children. It can cause different severity within the same family, and it can also cause structural disease or functional disease. So plumbing problems, but also problems with the way the heart contracts and relaxes. So we very rapidly came to the conclusion that this was not a pathway that was going to yield clinically relevant information that we could pass on to expecting parents and people planning families. There were big hopes for this thing called genome-wide association studies because we thought, well, congenital heart disease is a reasonably common disease and common variants should underlie common diseases. The short answer is it got us nowhere. And then enter stage left the new approach to gene sequencing, so-called massively parallel sequencing, which allows sequencing of all the coding and non-coding information in the human genome. And the first application of this study showed that in actual fact there were a lot of new mutations. When I say mutations, that's a bad word. These are just variants. These are different ways that the gene has been put together, and it codes protein differently. Lots and lots of new variants, some of which can be inherited, and so there is some relevance to this finding. But one of the downsides of this approach is that it's really just a research tool at the moment. There's a lot of unwanted information that you can get, including the possibility of finding out at the age of minus 20 weeks that the child has a breast cancer susceptibility gene. This is information that we really don't want to be getting into at this point. I know that sometimes more information is better, but not that kind of information at a time that parents are handling other information. There's also a lot of assumptions that are made in the analysis of this genetic information. So what we did here was to try and narrow down the scope of what was being examined. And we took a group of families that we knew very well who had lots of congenital heart disease in their family, but their problem had not been solved with routine clinical genetic approaches. So they've seen geneticists, and despite that, are without a unifying diagnosis and without an ability to understand whether it would be passed on to their offspring. Short story is, of those 16 families, we were able to identify about just over a third of the families. We were able to tell them what the cause of the heart disease was in that family. So this is quite a breaking story. It's a breaking story because this level of this diagnostic pickup rate is previously unheard of when approaching families who we didn't have answers for before. It's also reasonably straightforward to do using a list of genes that we put together amongst friends. There is, of course, still a lot of, there's the other 60% of patients who are not sorted, 60 to 70%, and there are further research approaches that may yield a result there. This is not the answer, it's an answer on the way to a better story, and it's an answer on the way towards being able to provide personalised genetic counselling. And you can see the generations involved in the study there. The future is pretty exciting, and recently in New South Wales there's a big push that every child that's born should have a whole genome sequence done at birth. Not surprisingly, this is being promoted by the people who run the machine, but it's an idea which in the era of personalised medicine is really gaining some traction. And I wouldn't be surprised if this in an experimental format was introduced in the next few years, because it's reasonably cheap, the potential benefits are huge, and the ability to go back and look at your information depending on what problems you have through life, whether, if you've got problems with attention or focus, let's look at the intellectual disability and autism genes. If you've got problems with ischemic heart disease, well, let's go and have a look at those genes. You can always refer back to your original sequence genome. It's exciting, but we're not yet at the point where you would be able to provide pre-implantation information, which I think is the point at which many families would say this genetic research, it's really making a difference to the decisions we make. In the 1.5 minutes left, I'm just going to talk about some of the exciting things that you should be aware of. The first one is the technology called IPS, Inducible Pluripotential Cells. A number of people won a Nobel Prize for developing this process where you can take a skin cell from somebody in the right circumstances. Cultured skin cells can be reverted to a multi-potential cell that can turn into everything from a brain cell to a heart cell and everything in between. This is a series of pictures doing exactly that, where the destination has been a heart cell. And this comes from a skin biopsy of a family, of some parents that were enrolled in our IPS study here and grown up by Richard Harvey at the Victor Chang Research Institute. At the moment, we're using this only to understand the molecular causes of congenital heart disease. But around the world, people are using iPS-generated cells as therapy. And the great thing about this is that modification of the cells might also involve getting rid of the problem that caused congenital heart disease in the beginning. And if you can grow a heart cell that doesn't have the functional problem that your heart might have, well, that's a really useful intervention, and we're getting a lot closer to that. It's a very rapidly emerging story. Similarly, on the stem cell theme, you will have heard about people with ischemic heart disease, heart attacks in adult life due to blocked arteries, receiving stem cell therapy to improve the function, particularly around where the heart muscle has been damaged. This technology in a different format is coming to congenital heart disease. You might recognise in the author list here Sunji Sano, who's a leading hypoplastic left heart surgeon, amongst other things, from Japan. And this group has used stem cells grown up from a little biopsy of the atrium at the first operation in the sequence for hypoplastic left heart, and then modified those cells and then re-injected them through the coronary arteries, and have shown that in the patients who are struggling with tricuspid regurgitation and poor ventricular function actually have better heart function, less tricuspid regurgitation, and actually have a much better growth velocity, which is a really powerful outcome for this phase 1 study, which is now being rolled out to further centres. This is very exciting work. We shouldn't forget the role of mechanical support, and I mentioned in my first talk today about the possibility of things to just give that venous blood a little bit of a push to reduce the venous pressure in the lower body just by a few millimetres of mercury. And there's all sorts of clever things that are being developed there. The other thing is that miniaturization of essentially adult ventricular assist devices, which have previously been used in the context of transition to or bridge to heart transplant, may actually be quite effective as a destination therapy, or at least to bridge children until adulthood. This device, which is the silver object there, that's just smaller than a lipstick. And so this is a device that's really well suited to pediatric practice. And the impeller that makes the blood go around is that thing that's sitting on the person's finger. This is exciting stuff. None of this is possible without a big team. I'm happy to answer questions about the future, but I don't really know what's out there, so I'll do my best. Thank you.